Hello, everybody. So I'm Sten Sörensen, CEO of Cereno Scientific. The company is formed out of very interesting research at Sahlgrenska Hospital, University Hospital, and the Wallenberg Laboratory there. Professor Sverker Järn and his colleagues have made some exciting findings around the biggest killer on the planet, thrombosis. And I'll present to you what we're about and how our development is going. So we are about to develop a new pharmaceutical drug for the prevention of thrombosis, as I mentioned, the biggest killer on the planet. And we are approaching this in a very different way than the major sellers and drugs in the market. Now, if you look at the market and you look at the causes of death, non-communicable death, you can see from the red staple here that almost half of the deaths in the world is due to cardiovascular incidents. And if you look to the right, you see the bluish pie parts of the pie there make up most of the pie chart and that's related to myocardial infarction and stroke incidents, so in the brain or in the heart. So, and that's thrombosis. So thrombosis is causing most deaths on the planet, and the key organs that are affected is the heart and the brain. Now, if you look at the market, you can see here that it's about to double in 7 years. And it's huge, because it's the biggest cause of death on the planet. Of course, there are drugs to try to alleviate that. And so the market is now $20 billion a year, and it's about to grow to 41 billion by 2024. And this is the market we are aiming towards. Now, looking at thrombosis again, every other second somebody dies of thrombosis. But more than 10 times that survive and are affected by this thrombosis in the brain or the heart or in the lungs or veins. And you can get a heart failure development in your heart, or you get an impaired brain function. So it's serious, and it is a big burden on the hospitals and economic society beyond the patients themselves. Now, the drugs that are used are called blood-thinning drugs. And I'm sure you have some in your network or in your family relatives that have experienced this, an infarct or a stroke. And they are given then blood-thinning drugs as a follow-up therapy. The problem, the drugs are good in the sense that they prevent thrombosis from happening, and there are older and newer classes. But they're all acting to weaken the coagulation system. And why is that a bad thing? Well, we have the coagulation system to stop bleeding. And if you weaken that, you are at higher risk of bleedings. And that's all around in your body. And you don't want to experience bleeds in your brain or in your stomach and elsewhere. So although they are very good at preventing thrombosis, you are at constant risk of experiencing a bleed, a severe bleed or any kind of bleed. Now, so the pharma industry has developed these drugs in moderate dosages. So they have an effect, but if you go up on the strength of the drug, you increase the bleeding risk. So they could have better effects if they could be dosed higher. So there is a need in the market for something that doesn't cause bleeds or the risk of bleed. And there are also need in the market, this huge market for better efficacy. So we can avoid these things to happen. If you look at clinical material, about 2.5–3% of all that are in the clinical materials with the latest drugs, NOACS, experience severe bleeds. Mostly in the stomach, but elsewhere. And up to 25% get any kind of bleed. So that would be, for instance, a nosebleed that doesn't stop. You have to go to the hospital and try to stop it. So, and the cardiologists and treating physicians all know about this, and they are worried about this side effect. And sometimes they avoid giving the preventive therapy because of the risk. Because an infarct, that's God's doing, but the bleeding is the doctor's doing, so to speak. So looking at you can see here is like a balance. And that's an illustration of how the body works with the coagulation system. So the body has a coagulation system to stop bleeding. So if you get a cut on your hand, you can see that there's a small blood clot. But the body also has a fibrinolytic system to dissolve blood clots. And that system is inside all of your vessels that each of you have, in the brain, in the lungs, in the heart, and elsewhere. And that's a system, a safety system for the body, because you don't want the blood to stop flowing. If it stops flowing in your heart, all the cells downstream die, and that's an infarction. And you might die yourself, or you get heart failure development, for instance. So we have this system, and if you look at this roof, you might see sprinklers somewhere. And you'd like that to be effective. If the doors were locked and you were in here and there's a fire, you'd like there to be water in the system. Keep that in mind. If there isn't water, it would be serious. And let's look at the body and how the body takes care of this. But first, if you look at this balance weight, you can see to the left that all drugs on the market today, the $20 billion market today, is addressing the coagulation system, the antiplatelet and the coagulation system to the left. Cereno is working to the right. We are working to optimize the body's own defense system, our own defense system. Okay, so let's look at how this can be illustrated. If this is a vessel that's cut in half, so to speak, and you see the bottom of the vessel, and that's a plaque. So atherosclerosis, in development, you get plaques. And as you age, there's a risk of them cracking, and then there's a bleed. Now, if that happens, there are signal substances that go up to the inner cells, the endothelial cell layer, and there is stored a protein called TPA, and that's released from that storage through this signal. And that's a fibrinolytic protein. So it helps to prevent the beginning blood clots from forming. So it stops at coagulation, but doesn't grow and occlude the vessel. It's a very important defense mechanism. There's another hormone in the body, in the blood, that's floating around, and that's there. It's called pi. It's there to take care of the TPA that's not used. Now, in risk patients, the TPA is not available. It's very low in the endothelial cell, in the small bags, granular, that it's stored. And pi is very high, especially in the morning hours when there's the frequent myocardial infarction occurs. So in risk patients, this system is weakened. Now, if you illustrate this with the heart and you cut the vessels the other way, you can see to the left there is a healthy patient, a healthy person. So there is a crack of the atherosclerosis. The blood starts to stream out and starts to coagulate, and TPA is showered out to stop it from occluding. If you look to the right, that's a risk patient. That's a risk individual. And risk individuals are those with that are obese, have high blood pressure, diabetes, and have had a previous infarct, and so on. In those patients, there isn't this water in the sprinkler. There isn't the water. So there is a high risk of the blood clots from growing and occluding the blood flow. And that's an infarct. And you can see from the heart that it's dying down there. Now, what is Cereno doing about this? Well, at Wallenberg Laboratory, they focused on trying to develop a drug that supports the defense system and actually restores it so it's powerful and can protect. And we call the drug CS1 that we're developing. It's actually the same substance as Johan was presenting earlier related to cancer, valproic acid or sodium valproate. When this is used, TPA is through an epigenetic mechanism produced in much higher concentrations and stored. So you restore the storage of the water, so to speak. And also pi is reduced. So there's a double positive mechanism. I'll show you what we've found. So CS1 is both increasing the synthesis for the storage in the vessel, but also increasing the release of it. And at the same time reducing the main blocker of TPA, which is pi. What is the advantage with this? Well, as you heard before, there's a risk if you address the coagulation system. This doesn't. This works on the fibrinolytic system. It only builds up the defense system for the body. And it's only activated, as you saw, by the body itself to protect itself. Now, what I'll jump over to what we have seen, what we have documented. So we have looked in animals that have been given this substance for the TPA production. Do we affect production? As you can see here, this is in mice, in aorta in mice. To the left is untreated, and to the right is those that have received the substance. And the green is TPA that's stored in the endothelial cells. So it's a high improvement of TPA being stored. This is the proof of concept in live animals study that was published a couple of years ago. And you can see to the left, these are mice that have been given the drug 96 hours in advance, so 3 days in advance. And the red line is the treated mice, and the black is on the control. And as you can see, already after 2 minutes, the red line is flat, more or less. And it shows the buildup of fibrogen, or the thrombosis size, in the vessel. And you can see on the photograph to the right that the thrombosis is much smaller if they are pre-treated with the substance. These are experiments in man, and the left slide shows that the substance increased TPA release in the forearm model. And the right slide shows that there is a 50% reduction of pi in blood plasma. So we affect 2 key markers for myocardial infarction or thrombosis in man. Now, is there any other indication that this works in man? Well, in 2011, a large epidemiological study from Denmark, the whole Danish population, 150,000 individuals were studied for 10 years retrospectively, and what epileptic drug they were on. And in only one group, there was a difference in cardiovascular events. And that's what those who were treated with this substance, which has been used for epilepsy for 45 years. So there was a 40% less myocardial infarction in those epileptic patients that got this drug over this 10-year period. There was also a tendency for reduced stroke. Now, what have we done? Well, we have taken this substance and we have reformulated it into something that will fit the patient category to address these 2 markers, the pi and the TPA. And we have worked with Galenica, a Malmö company, for formulation. And we put it into a study in volunteers or risk individuals to look for safety, tolerability, and pharmacokinetics. And also if we could affect pi, we measured. And we showed, actually, we got all the findings we wanted. The formulation that we have developed is very advanced. It's a timed release, a timed immediate release formulation to address the pi concentration in the morning hours. Now, so we're moving into phase 2, and if you want to do phase 2, you need money. And we secured a financing agreement with a fund in this spring of 82 to 106 million Swedish that will bring us through phase 2. And over the last 8 months, we have had procurement of CRO who should do this study for us. The study that we will document that we can prevent thrombosis in man. And we ended up signing with the OCT group based in St. Petersburg and operating in Europe and US. And we will run a study in 20 clinics in Russia and Bulgaria in orthopedic surgery. And in orthopedic surgery, there's a fairly high risk of getting thrombosis. So that's a very good model to use. Now, I should mention, I'm running out of time, but I should mention that we got very positive news now in September that we got notice of patent allowance for the US market, which makes up half of the 20 billion that I just mentioned. Let's jump over. So for this year has been a really good year for Cereno because we have finished our phase 1 trial. We got this data that we wanted. We have financed our phase 2. We got US patent allowance for the biggest market in the world. And we signed the CRO group for the phase 2. So we're moving ahead. And the milestones coming up is that we're scaling up our CS1 product for the clinical trial. And we are submitting our documents to authorities by turn of the year. And then we're going to start the phase 2 trial next year. All right, hope you found it interesting.
Thank you, Sten. I find it really interesting for me. So let me get this straight. You're moving into phase 2. You have a drug that is a completely new concept, treating thrombosis, preventing thrombosis. And a quarter of the patients today on thrombosis prevention drugs are at the risk of, or they experience bleeds. You're valued at like 37 million Swedish kronor. How's that possible?
Yeah, good question. So I noticed Fredrik got the question about the economic prospects or financing prospect in the market. And I have an issue with the financing market in Sweden compared to the US. I think that there's of course the dynamics are very different, but if you look at the valuation of new biotechs in the US, they are averaged between $150 and $350 million when they go on IPO. They might do that later, but still. So I think if you are in phase 2, like we are, you would be valued maybe 10, maybe even 20 times higher if you were in the US. Now, there might be other reasons for our valuation, and that's actually our financing structure that we have.
Yeah. You also are in this convertible loans deal.
Exactly.
Yeah. Well,
do you want me to comment on that?
Sorry.
Yeah.
No. Yeah. Why not?
Yeah. So we signed the deal with a European High Growth Opportunity Securitization Fund to be sure that we had the capital for our phase 2 program. Now the downside with that is that the fund is selling the shares on the market continuously. So as we pull down our convertible loans, they can convert and sell on the market, and that has put a downward pressure on our pricing. So if you're interested in Cereno, you have to sit in there for the phase 2 trial results, which is what we are aiming for. We look at the technology, but we would like to be valued on the technology basis rather than retail, actually.
Yeah. Then again, going forward, then coming into phase 2, and as you go forward and towards these results that you're hoping for, and I'm sure you expect them to be positive as well, that would then be quite an opportunity to grab a stake in a company that's really low valued in such a late phase. Okay, let's not speculate. I know you can't speculate on that. So, I mean, the market is projected to grow to like $40 billion globally in 2024. Say that your concept really works in man, you get statistical significance and everything is fine. What kind of market share is realistic to hope for in this kind of market?
I'll have to come back to you with that. We're actually looking at different scenarios at the moment. I think we will, if we continue and pursue our phase 3 program ourselves, we'll probably hit the market around the time of, or a little later than the time of that forecast. So if there is a $40 billion market and we have a drug that's first in class and doesn't cause bleeds and is effective, you know, it's going to be worthwhile.
Going to be worth the wait. Thank you very much, Sten, for coming today. Give
Thank you.
A warm hand.